This is help me video for question 4, the essay. Genes consist of introns and exons, so we need to know what those are. Introns do not code, whereas exons do code, so it tells us that, that's okay, you knew that anyway. Introns are removed prior to messenger RNA leaving the nuclear pore. Okay, it's just telling us what we know from the theory. A messenger RNA base sequence is shown below. The intron is in bold, and we've got some changes and mutations. So we've got some mutations. What have we got? We've got that's a mutation there, and we've got one that you might not have spotted there. So 2 mutations. So one of these mutations is in the intron, and one of them is in the exon. So if we change the colour of the exon there. So these are 2 different types of mutation, one in intron, one in exon. Then we've got some information. We've got the messenger RNA codons in a table. So what we've got to do, using the information above, illustrate how the genetic code results in a protein of the correct shape and the impact of a change in the base sequence. So things we're going to have to talk about. We need to talk about transcription first of all. So transcription. We'll talk about translation. And then we'll talk about these mutations. Okay, so in transcription we need to talk about we've got our I can't even draw DNA. Okay, so we've got our DNA. So we've got to go from our DNA. We're going to produce this pre-messenger RNA. Then we're going to produce our messenger RNA. And in there, that's this area here that we're talking about, the splicing. So we're going to talk about what happens to DNA. So you're going to talk about any enzymes, such as maybe helicase, RNA polymerase, unzipping, unwinding. We're going to mention, so when we've got this unraveled, we're going to talk about templates, what we're making our messenger RNA out. We're going to make it out of nucleotides, so RNA nucleotides. How will they attach? We're going to talk about complementary base pairing, and we've got no T's, remember, so it would be A to U, C to G, or indeed T to A from our DNA. And then you can talk about the splicing, the removal of the introns, removal of introns, and then leaving the nucleus by the nuclear pore, like it mentions there. Then all the usual stuff for translation. So talk about the messenger RNA codons, ribosomes, what happens there. So two codons are held together. Of course, we'll talk about tRNA and anticodons and so on. You could give examples, maybe from what you've been given there. Maybe you give the AUG and the UUU. So e.g., you could tell us what some of these might give you. And then the actual meat of the essay is the mutations. We can talk about the exon mutation, exon mutation, and you could tell us exactly what would happen. So tell us the change. So this one will give a different amino acid, so a different amino acid, and then you can give the effect on the protein, the change in shape and the change in the function. So shape and possible change in function. Maybe come up with some examples, like receptors and shapes like that, hormones. And then we've got our other mutation, which is our intron mutation, which is our UAA. And the effect on that, well, because that's going to get cut out, that's going to have no effect. It's going to be a silent mutation. So you are realizing that yes, there is a mutation there, but it's not going to make much of a difference. So all of your knowledge of transcription, translation, and eventual shaping of the final protein, and these two mutations, one that will have an effect and you can describe that, and one that won't have much of an effect.